Today I'll be presenting the Gut Microbiome Lab by Ify, Mediz, Harlicula, and myself, Tahara. We are Section 11, Group E. Molecular biologist Joshua Lederberg has defined the gut microbiome as the totality of the things within the gut microbiome. This can include the bacteria, the viruses, the fungi, the protozoa. All of those make up the gut microbiome and their collective genetic presence in the GI tract or the gastrointestinal tract. The gut microbiome is composed of bacteria, commensal and pathogenic, which resides in the GIT, as I have mentioned previously. In the past decade, the gut microbiome has been explored for potential gut microbiome host interactions. In the past decade, the gut microbiome has been looked at to see its effects on the immune system or its neurological effects. The gut microbiome plays an important role in nutrition and mineral absorption, synthesis of enzymes, vitamins, amino acids, and the production of short fatty acids. So many researchers did a study to see if the gut microbiome between a lean person and an obese person was different. In order to do this study, they analyzed the difference between lean and healthy individuals and obese individuals. They specifically tested the bacteroidetes and the firmicute phyli and found out that there was no difference in the gut microbiome between an obese person and a person that was lean. According to their t-test, the p-test, and the bacteroidetes phyla, it came out to be 0.30, while the p-value of the phyla was 0.86. This concluded that obesity is not associated. So the gut microbiome affects different areas of the body, such as growth and development. This can include parts of the nervous system. Many researchers did a study to find out if there was a connection between the gut microbiome and autism. They concluded that there is an effect between gut microbiome and autism due to the influences of neural development. To come to this conclusion, the researchers conducted a study. This study tested to see if the antibiotic-treated SPF mice as well as the germ-free mice would be affected. In this study, they concluded that the behavioral effect of antibiotic treatment did not occur in the GF mice or the germ-free mice, suggesting that a colonized gut was required for the effect of autism. For our materials and methods, there is a comparison of our animals. These animals, Hoshi and Nova, are the animals from Group B. The animals that we had were Magic and Nala. As you can see from Magic and Nala, they are both the same breed, which are pit bulls. However, their age difference is very vast. One is male, one is female, and they're both unaltered and have very good condition. So in order to find the purification of DNA, we use a nanodrop UV absorbance spectrophotometer. This is used at a wavelength of 260 to 280 nanometers. Whenever we use this, we use the Kaizen kit model. We made sure that we followed the Kaizen kit model exactly to the instructions with no change in the protocol, as you can see here. And we found this from the Lab Manual Gut Microbiome 1. In order to do the quantification of the DNA, we had to use the quantitative polymerase chain reaction, or qPCR. This was used to amplify the bacterial genome 16S RNA gene. The nanodrop spectrophotometer that we talked about in the purification was also used here with the same 260/280 nanometer ratio. So for our qPCR, we use 4 different ones. For the ones that we use the N-actinobacteria. We use this because it is a major phyla in the soil systems. Our next one that we use is firmicutes. We use this because it is found in plants and includes some pathogens like in endspores that can form endospores. And the Videocraft, we use this because it can cause a change in the gut microbiome that can lead to cancer. And lastly, we use the acetobacteria because it includes acetophilus. Acetophilus is found in archaea, bacteria, and eukarya. We found the results for our DNA quantification results. For all our other samples, except for our test subject 672, everything was between the range of 74 and about 80. Sample 672 is the outlier. As you can see, its concentration is 101.8. Again, our samples, the AZ60 was the same for all our other three, except for 672 as the outlier. When we get over to the absorbance level, 816 is the outlier with a very high absorbance of 2.05, and all the other test subjects had roughly around the same absorbance. Control, we found that while doing our phyla, we did not get a result. This means that we did something correct because you're not supposed to get a result whenever you do your phyla 1. But as you can see here, we have a lot of varying numbers. For phyla 1, the anti-log, for our test subject 816, it was 3.46. For our test subject 1057, it was 2.28. For our test subject 671, it was 1.22. And for our test subject 672, it was 1.25. Oh, sorry, I'm not ready. Upon looking at our results from the qPCR, which is the actinobacteria versus the Microbiome, we can see that the positive control is very close in numbers to our 816, which should not be happening. However, our positive control was 1 for the anti-log, which means that we did something correct because the positive control should be very low. And then here's some of our results. So for 1057, we could not get a result for phyla 1 CQ, and also for the anti-log since we didn't have a phyla 1. But all the other numbers, although our test subjects seem to be within the same range, except for 816, which is the outlier for the phyla 1 CQ, being at 22.23. At the end of our phyla 1 antilog, phyla 2 antilog, we found that 816 is 0.6518, our test subject 671 is 1.35, and our test subject 672 is 0.0380. Of the qPCR, firmicutes versus acinobacteria, we did not do a positive control, so there's not one here. But as we can see, majority of our test subjects seem to be within the same range for the phyla 1 CQ. When we do the phyla 1 CQ antilog, we can see that there is a significant difference between 1057 and the rest of the test subjects. For our gut microbiome, we determined that the DNA extraction was successful in terms of quantity. There is enough DNA extract to continue. Sample 816 was the lowest. Samples 1057 and 671 were relatively close. They were between 79.6 and 79.9 microliters, respectively. And sample 672 had the highest concentration of 101.8. qPCR run, the bacterial positive was 11.52. This is under 15, which is low. So that means that it is correct because we can continue with it. However, the fungal positive control had a CQ of 33.38. This is way too high. It should be less than 25. The bacterial never control was 27.29, which means that the DNA was effectively absent. The fungal never control was not detectable at all. In our second round of qPCR, we found that the negative control for CQ was 27.25 for the phyla 1 and 30.49 for the phyla 2. In both of these cases, the DNA is effectively absent. For the positive control, we found that the CQs were 26.57 for phyla 1 and 28.02 for phyla 2. These values are way too high, which means that there is no DNA that is present. In our third run of the qPCR, we found that the negative control was 31.49. This indicates that it is high enough to qPCR, our sample 816 had about 51.7, while sample 1057 had about a ratio of 2.30. They have several degrees of separation, and this indicates that the gut microbiomes are different. For the second qPCR, sample 816 had a ratio of about 0.65, and sample 1057 was undetectable. For the third qPCR run, we found that sample 816 had a ratio of about 1.0, and sample 1057 was not detectable. Our experiment found that sample 816 consistently had a higher presence of organisms in the microbiome. While while we can assume that this is caused by biological sex, we are not certain. To better improve our experiment, we should have picked puppies that are from the same litter so that we would know where they're coming from and if their sex, to determine their sex, their parental lineage, things of that nature, diet, and genetic factors.